What is EDNA Metabarcoding? Have you ever wondered how scientists can identify various species in the polar regions without directly observing them? The answer lies in a fascinating technique called Environmental Deoxyribonucleic Acid Metabarcoding. This method allows researchers to detect and identify multiple species present in an environment by analyzing tiny fragments of DNA found in environmental samples like water, soil, or snow. Instead of capturing or observing organisms, scientists collect samples that contain traces of genetic material left behind by these organisms, such as skin cells, hair, or waste. In the polar regions, this technique is especially useful for monitoring biodiversity and ecological changes in these remote and harsh environments. Observing or capturing species like polar bears or marine organisms can be challenging and disruptive. Environmental deoxyribonucleic acid metabarcoding provides a non-invasive alternative that can yield detailed information about species presence, population genetics, and even diet. The process begins with collecting environmental samples, such as water from Arctic seas or snow from polar bear tracks. These samples contain tiny amounts of DNA shed by organisms. Scientists extract this DNA and use metabarcoding, which involves amplifying and sequencing specific genetic markers. These markers are short DNA regions common across many species but variable enough to distinguish them. By comparing the sequences to reference databases, researchers can identify the species present in the sample. In polar research, environmental deoxyribonucleic acid metabarcoding has been successfully applied in several ways. For instance, scientists have detected and genotyped polar bears from snow tracks without disturbing the animals. They scrape snow from footprints and analyze the DNA left behind. This method allows researchers to monitor polar bear populations and gain insights into their diet and movement patterns with minimal impact on the animals. Additionally, researchers assess biodiversity in Arctic marine ecosystems by extracting environmental deoxyribonucleic acid from seawater or sediment samples. This helps track changes in species composition related to environmental factors like temperature shifts and sea ice cover, which are critical for understanding the effects of climate change. Another application is monitoring shifts in community composition over time through repeated environmental deoxyribonucleic acid sampling. This provides data on how warming and other environmental drivers affect Arctic flora and fauna. The technology used for collecting environmental deoxyribonucleic acid samples in polar regions often involves specialized equipment designed to operate in extreme cold and remote locations. For example, researchers may use Niskin bottles to collect water samples from research vessels. These samples are then filtered and preserved under sterile conditions for later laboratory analysis. Snow samples from animal tracks are carefully collected and preserved to maintain DNA integrity. Overall, environmental deoxyribonucleic acid metabarcoding offers a powerful tool for polar research. It enables comprehensive, non-invasive biodiversity assessments and ecological monitoring that are well suited to the challenging and sensitive polar environment. This method complements traditional research techniques by providing additional data without the need for direct contact with wildlife or extensive field sampling, which can be difficult in polar conditions.